questions on that pre-midterm. So if you don't understand something, you have questions, come to that review session. Get all your questions answered at that point. Oh no, what just happened? Okay. Did the internet go out?
No, internet looks good.
All right, let me see.
It flashed back up for a second.
Did it? Oh, all right. Let's hope that stays there. That was strange. All right, so yes. And so then finally, the midterm itself will be on Tuesday, October 28th, right here in class. You'll again have 80 minutes. It will also be multiple choice, but it will be on paper, and you'll have bubble sheets to fill out for that. Question? It will not be on Zoom, but we have scheduled it to be podcast recorded. We cannot control IT and know on a Sunday when that podcast will actually post, if it will do it correctly and all of that. We will do our best to make sure that happens, but try your best to be here in person. But otherwise, we have scheduled it to be podcast. Other questions? Yes?
The review exam on the pre-midterm on Kansas, is it one time we can only take it and then we review our answers afterwards?
So it will be one time, and after the window that has been open has closed entirely, then your answers you'll be able to see what you got right or wrong on that. You'll be able to see your score. Other questions? All right, make sure you schedule your time to take that pre-midterm. And also, make sure that you can take a Canvas quiz using your browser. Some of you have taken advantage of this practice online exam. Most of you have not. Okay, so let's review what we talked about in class last week. So we started off by talking about this method we can use in the laboratory to amplify a specific sequence of DNA. This is very useful in forensics, for DNA fingerprinting, and it's called the polymerase chain reaction, or PCR. So if we're going to perform a PCR reaction, what are the 4 basic reagents we need to put into our test tube? What's one of them? Yes? A primer, yeah. We need primers because we're doing DNA synthesis. What else do we need? Yes? Yeah, we need Taq polymerase. It's this special thermal stable DNA polymerase. What else? Yes? We need a DNA template. What's going to be copied, right? And what's the final thing we need? The dNTPs, the deoxynucleotide triphosphates. So we mix those all up, and then we put it into a machine called a thermal cycler that's going to go through these 3 basic steps over and over again. So the very first step, if we're going to copy our DNA template, we need to denature it. We need it to become single-stranded. And what the machine will do is it'll just use high temperature to break all those hydrogen bonds, and now you have single-stranded DNA templates. The second step in the PCR reaction is going to be able to cool the temperature down to a level where your primers will be able to base pair. And you want to design your primers so that they will amplify a specific region of the DNA, so you make sure they're not going to base pair all over the genome. And you want to make sure the 3' ends are facing each other, because that's the direction you want synthesis to go in. So after the primers have annealed in step 2, we then have step 3, where our Taq DNA polymerase is going to use those deoxynucleotide triphosphates to add onto the 3' hydroxyl and synthesize DNA using the DNA template, always synthesizing in the 5' to 3' direction. And then that's going to repeat, and if you repeat this cycle about 20 times, you get a million-fold amplification. Any questions about PCR? All right, let's do a clicker question. So which of the following primers could be used in PCR to amplify the entire sequence shown below? And then note that only the first 3 nucleotides of the primers are shown. So we want to amplify this entire sequence. I think something is wrong with, hold on a second. Okay, it's recording, you guys. Oh. Hey, hey, hey. The base is not working? All right. Let's... Huh. It's taking responses, but it's not... All right, let me just... All right. Sorry, I'm going to like just take it all down and put it all back up again because there's things that are not working. Still not reading the base, is it? Try one more time. All right, well, I don't know why the base will not read this time, so Let's just keep going with class, and I'll figure out what to do about your extra credit for clicker questions today, okay? So most importantly, let's just go through this. All right, so sorry it's been flashing up and off, and hopefully you've had a chance to look at this question and figure out what are the first 3 nucleotides, the primers that you would use for this. Oh, I see something else weird that's happening here. Hold on. All right, so who wants to share their answers for this question? How would you design your primers for this? Quick, before it disappears. Any ideas? Or just tell me your thought process of how you would go through this. Okay, here's a sequence you want to entirely amplify, so you need to design primers. So what do you want to think about when you design your primers? Sure. Yes, exactly. So we want to make sure that when our primers base pair to the template, the 3' ends are facing each other. So based on that, what is a good option here? I don't know what the majority of you picked. Really sad. This is not working. No one wants to venture an answer? All right, so maybe it'll be helpful to think about it this way. Oh, why did it do that? All right, so when you have the DNA and it is going to be denatured, the first step, it originally was double-stranded, right? So if it's easier for yourself, just write the bottom strand, make it double-stranded DNA, and then imagine it denaturing, and then how would you get those primers to base pair? Like this, right? So you just denature the DNA, and so on the bottom strand, your synthesis is going to be in the 5' to 3' direction. So the sequence of this primer is actually going to be exactly the same sequence as that top strand going in the 5' to 3' direction. Here, we also need to have synthesis going in the 5' to 3' direction. So we are going to have our primer base pair to the top strand, which is what was shown. But if you wrote yourself the bottom strand here, again, it's going to be exactly that same sequence. And so that made the answer then be B. Does anyone have any questions about designing the primers? Does this make sense to you? I think it's often easiest, especially when you're first doing this, just write the other strand. If I give you one sequence of DNA, you know what the other strand is going to be, and then you can imagine it denaturing and the primers you're going to need for synthesis. Okay. So then after we learned about PCR, we talked about DNA repair mechanisms, because our DNA is always being damaged all the time. Even when it's being copied by our high-fidelity DNA replication machinery, every now and then a mistake is made. And so we need a lot of mechanisms in order to deal with all these damaging events that can happen to change the sequence of our DNA. It's dangerous to have sequence changes within our DNA. And so we talked a lot about the DNA repair mechanisms that can be used to fix DNA. The first thing we actually talked about is if you even have your high-fidelity DNA polymerases, which ones are those? Epsilon and delta, right. So if they're zipping along doing leading and lagging strand synthesis, every now and then they incorporate a nucleotide that is the incorrect base pairing. And they are actually able to recognize that, and they can pause, clip out that wrong nucleotide, put in the right nucleotide, and continue going. That is their proofreading ability. But sometimes they're moving so fast, they miss that mistake. But we have another mechanism that is actually active during DNA replication that has factors sort of checking what the polymerases have done to find any mismatches in them. And this is a repair mechanism called DNA mismatch repair. Which one, 2, or 3, is diagramming mismatch repair? 2, yes. Number 2 right here. Because what happens in mismatch repair is the incorrect nucleotide, the newly incorporated nucleotide, it's recognized that that's in the daughter strand DNA. That is recognized, the DNA is unwound, and nucleases take out that incorrect nucleotide and the surrounding nucleotides. And then your high-fidelity DNA polymerases see that 3' hydroxyl, looks like a primer, and it can just add dNTPs using the other strand as a template until it reaches this 5' end here, and then ligase will seal that gap. That is DNA mismatch repair. But many times our cells are not dividing, replication's not going on, but there are simple byproducts of metabolism, deamination events that can change the sequences in our DNA. We get base changes within our DNA. And so those need to be fixed. So this GT mismatch right here, this T was probably a methyl C, and so there is a repair mechanism that's going to remove just that base. What is the name of this repair mechanism? Base excision repair, right? It'll excise that base and in fact the entire nucleotide. And then there's a special DNA polymerase that does base excision repair. And which one is that? Beta. DNA polymerase beta puts in the correct nucleotide, ligase seals the gap. And finally, sometimes there are much more abnormal changes to our DNA. They cause these bulky lesions. They distort the DNA, such as these thymine-thymine dimers. It's very easy for the repair mechanism to recognize this and excise the incorrect nucleotides as well as surrounding nucleotides. What is the name of this repair mechanism? Excision. Nucleotide excision repair. So that area is removed, and then your high-fidelity polymerases fill in using the undamaged strand as the template. All of these single-stranded DNA repair mechanisms are considered to be air-free. And the reason they're air-free is the other undamaged strand serves as a template. So there is a template to know what sequences to put in. Oh my goodness. Okay, sorry. All right, but then there can also be double-stranded breaks in our DNA. And we have 2 general repair mechanisms that can fix a double-stranded break. Which one of these 2 choices is essentially air-free because a template is found? Number 1, which is, homologous recombination. Unfortunately, which is the repair mechanism that is much, much more commonly used because it is quicker and more efficient because we often don't have a template nearby? Number 2, end joining, exactly. And we have a clicker question, but it doesn't seem like it's going to work. I'll try. Oh, it's working on my base. All right, so if you click in now, you will get credit for clicking in previously. So please click in. Which of the following statements about double-stranded DNA repair mechanisms are correct? Maybe quickly click in to get that in case it disappears. It'll be recorded then, okay? You can always change your answer. Ten more seconds. right, looks like everyone has their answers in. Good, good, good. Let's see if we can see the answers. All right, so we have a majority voting for C. We have some people liking B as well. So let's go through these options. Who wants to volunteer their answer and reasoning? The technical glitches have thrown all of us off, right? Sure.
I chose B because I know you need an extra copy of the chromosome to actually use that as a template strand, and I'm pretty sure S phase is producing that extra copy.
All right, so B says repair by homologous recombination is only possible during S phase when DNA is being replicated. So you're right, when we do homologous recombination, we need a homologous chromosome. And if the DNA has been recently replicated, that sister chromatid, they're even going to be really close to each other, and you're going to get very efficient homologous recombination. But is that the only source of a homologous chromosome? What is the other option? We're diploid, right? Meaning you have 2 copies of almost all your chromosomes. So you can actually have the homologous chromosomes serve as a template for homologous recombination in cells that aren't undergoing DNA replication. Now that is going to be much less efficient because somehow that chromosome needs to find its homologous pair. But we know it happens. This does happen in vivo through mechanisms we don't entirely understand. But those are our 2 options for having a homologous template in order to do homologous recombination. So we have 2 options. It's not only during S phase. So B is not correct. What about A? A said end joining is error-free when it ligates 2 ends of the same chromosome back together. I'm seeing people say no. No, because what happens? As soon as your chromosomes break, nucleotides start to be chewed away. To actually even do the end joining, you need to make blunt ends, so the nucleases are going to chew some away. Sometimes nucleotides get added to those free ends, so it's almost always going to have errors introduced through end joining, even if it's the same chromosome coming back together. What happens if 2 different chromosomes come back together? What do we call that? A translocation, and that is definitely an error and a big change. All right, so A is not correct. What about C? So a lot of you like C. During repair by homologous recombination, 3' single-stranded overhangs are created on the broken DNA to serve as primers. Yes?
I said C because I was looking back at our last notes, and it says dsDNA rate is recognized in nucleotides and leaves the 3'.
Yeah, so it's almost exactly from the notes. Exactly. For this to happen, to get repair using the homologous chromosome, we need this 3' end to serve as a primer to invade the homologous chromosome base pair and then get added onto. So C, in fact, is a correct answer. So let's just look at D. D said during double-stranded DNA break repair, ligase is used for end joining, but not homologous recombination. No, because we need ligase for all of the DNA repair mechanisms we talked about. Any questions about that clicker question? All right. Okay, so we have already talked about our first major molecular mechanism, and that is DNA replication, the process of DNA being copied into more DNA. Today we're going to move into our second molecular mechanism, which is copying DNA into RNA. And so this process, of course, is called transcription. And the type of transcription we're going to talk about from now up until the midterm, we're always going to be talking about transcription of protein coding genes. And so this would be the synthesis of what we call messenger RNAs. So today we'll begin by talking about the general process of transcription, the 3 phases of initiation, elongation, and then finally termination. We'll review a little bit about how genes are differentially organized in prokaryotes versus eukaryotes. We'll then talk about a new type of experiment that we can use to detect whether an RNA has been produced. And then we'll talk about transcriptional regulation in bacteria. Okay, so just like when we talked about DNA replication, we need some basic reagents for RNA transcription to occur. What do you think one of those reagents is going to be for RNA transcription to occur? What do you need? You need a polymerase, you need proteins to carry out the work. You need a template, and is the template's going to be DNA, right? You're going to be copying DNA into RNA, and so what's the third thing you need? Yeah, ribonucleotide triphosphates. So there's some differences from replication, right? So we're using rNTPs or ribonucleotide triphosphates instead of deoxynucleotide triphosphates. And then also, I thought we needed 4 things when we did DNA replication. What was that 4th thing in DNA replication? A primer. Why don't we need a primer for RNA synthesis? Yeah, this RNA polymerase, this enzyme, it can start synthesis just by laying down nucleotide triphosphates, that base pair to the template, and doing synthesis. It doesn't need to add onto a free 3' hydroxyl. So we're going to have a DNA template. Synthesis, as always, whether you're DNA or RNA, is going to be 5' to 3'. So you're still going to be moving in the 5' to 3' direction. And the polymerase is going to take nucleotides, so ribose nucleotides, that will base pair to a sequence in the template. The preceding nucleotide 3' hydroxyl will attack the alpha phosphate on the incoming triphosphate in order to form this phosphodiester bond. And this is just zoomed in on that polymerization step. So the 3' hydroxyl is attacking here, pyrophosphate leaves. Okay, there's some terminology. Excuse me, I need a drink. So there's some terminology that we need to be aware of when we're talking about transcription in reference to where does transcription begin. So when we're talking about a gene and transcription, the site where transcription begins, the convention for that is called the +1 site. So it is the +1 nucleotide. And then all sequence that is transcribed, that as the polymerase moves downstream and is synthesizing RNA, is also going to be plus. So you just count up from +1, +2, +10, +30, going this way. Excuse me. All sequence that precedes the +1 site is going to have a negative number. And this is called the upstream sequence. This is where your core promoter sequences are going to be. What about, is there a 0? No, there is no 0. So the nucleotide that is right before the +1 right here is simply -1. So if it is before the transcription start site, it's going to have a negative number. If it is after the transcription where the polymerase is actually synthesizing RNA, the polymerase is moving down the template, it is the downstream sequence, it's going to be positive. Okay. When we are looking at the region that is being transcribed, we also have these terms non-template and the template strand. All right, we've already talked a lot about a template. So what is just a general definition for a template? How would you define a template? What is it? It's what gets copied. It's what gets copied. And the way that you copy it is you use your base pairing rules, right? So the template strand is what the polymerase is going to use to know which RNA nucleotides to put in. It is going to be what gets copied. That's why the other strand is called the non-template strand. It is the sequence that is not being copied, but you'll see something about the non-template strand. So if we are using this template strand to synthesize RNA in the 5' to 3' direction, moving downstream here, what is going to be the sequence of the first 3 nucleotides? What's the first thing you're going to write? 5', right? So 5', C, U, because we're making RNA, G, exactly. So This template strand is being copied, and it's just being copied using your base pairing rules. But look at this. If you look at that RNA sequence, what is it? It is actually the same exact sequence as the non-template strand, except for you have U's instead of T's. So, if you know what the template strand is, you know what the non-template strand in it is, the one that is complementary. And the non-template strand is simply going to be the same sequence as your RNA, except for U's instead of T's. Is that clear to everyone? So this terminology is important to understand when we are talking about synthesis and template versus non-template. All right, so let's review this again. So if we have our polymerase, it's going to sit down around the +1 sequence so it can start synthesis there. +1 means where we're starting synthesis. And as it moves down, copying the template strand, it is moving downstream, and all of those positions are going to be plus. Everything before the +1 is going to be negative, and that is then considered upstream. If I told you that there is a G at the +1 position in the template strand, what is going to be the first nucleotide in your RNA? C, exactly. If I tell you template, you know it's going to be what's complementary. If I tell you what the non-template sequence is, what are you going to tell? If I say it's A, it's going to be A. It's going to be exactly the same. Exactly. Very good. Okay, so let's talk about the different stages of transcription. So first we need to start, we need to initiate, and the way this generally happens is RNA polymerase, and when we're talking about RNA polymerase, this is actually a complex of proteins. You'll see in subsequent classes there's a lot of proteins involved in this process. But what you'll need to have is the RNA polymerase complex to bind to the promoter sequence around where the +1 start site is. And in order to begin synthesis, the DNA template needs to be unwound. Once you have that +1 site in the template exposed, you can then bring in ribonucleotide triphosphates to base pair to that +1 nucleotide in the template. And then at the +2 position, the next rNTP would come in, and the polymerase would catalyze a phosphodiester linkage between those first two rNTPs. Once you've catalyzed that first phosphodiester bond, you're now ready to move from the initiation phase into the elongation phase. And so elongation phase is just going to be the polymerase moving down the template, using that sequence to know what rNTPs to add to the RNA. Now don't get confused by this statement. It says polymerase advances 3' to 5' down the template strand. And that's true. That's the direction it's moving on the template strand, because synthesis is always 5' to 3', and we always need to be anti-complementary. Does that make sense? All right, so the polymerase is going to continue synthesizing until it's time to terminate. And termination is very different in prokaryotes versus eukaryotes, and depending on what type of gene you're talking about. And we'll talk about termination of protein coding genes after the midterm. But for now, just know that at some point, synthesis will stop, polymerase will leave the template, and you'll have your new completed RNA strand. So those are our 3 basic stages. You have to initiate, start synthesis, you then elongate, so continue synthesis, and then finally end or terminate. All right, so let's review a few differences about prokaryotes versus eukaryotic cells. So first of all, the 2 pictures I have here, the one over here further from me, is this a prokaryotic or eukaryotic cell? Prokaryotic. Why? How did you distinguish that? Yeah, there's not a nucleus. There's what they call a nucleoid here, but that's because that's where all the DNA is, right? Whereas our eukaryotic cells, we have this nice membrane-bound nucleus, other membrane-bound organelles. Now, when we're thinking about transcription, there's a few things that also distinguish prokaryotes versus eukaryotes. We need to consider how the DNA itself, right, that's the template that's going to get copied, how it's packaged in these 2 different cell types, how genes can be differentially arranged, as well as how messenger RNAs might need to undergo processing. So first thing, when we're talking about a prokaryote versus a eukaryote, what is the genome like in prokaryotes? Compared to the chromosomes we have in eukaryotes? Yes? They're circular. So do they have telomeres? No, they don't have linear ends, so there's no telomeres. So that's one major difference between prokaryotes and eukaryotes. What about, we spend a lot of time talking about the packaging of DNA in a eukaryotic cell. So how is our DNA packaged inside of eukaryotic cells? Histones, right. So histones are the proteins that form the nucleosomes that then wrap up further to make chromatin. Do bacteria have histones? No, but they do have proteins that do bind their DNA. They're just not histones in the same sense. So DNA is still packaged inside prokaryotes. It's just different types of proteins and arrangements. And so here's some of the things we already said. So in prokaryotes, one of the differences is a circular genome. You don't need telomeres. Eukaryotes, we do need these telomeres to protect the ends of our chromosomes. There are different types of proteins that package the DNA in these two cell types. Prokaryotes very often have their genes arranged as operons, as opposed to many eukaryotes that have separate genes. What do we mean by an operon? Does anyone know what an operon is? So it's a group of genes that are going to be synthesized together, and we'll look at an example in the next slide. Whereas many eukaryotes don't have this operon, but there are some eukaryotes that do have operon genes. And then, also in prokaryotes, actually, since there is no membrane separating a cytoplasm from a nucleoplasm, the translational machinery actually has access to the newly synthesized RNA as it's being made to rapidly translate proteins in bacteria cells, whereas in eukaryotes, the newly made messenger RNA needs to get out of the nucleus into the cytoplasm. To do that, there are different types of modifications and processing events that need to happen. We will go into detail about capping, splicing, and polyadenylation after the midterm. All right, so let's review what we mean by an operon. So the example shown here is the operon for making tryptophan. What is tryptophan? What is tryptophan? It's an amino acid, exactly. It's an amino acid. Bacteria have all the enzymes needed to make tryptophan. Can humans make tryptophan? No, actually we can't. It's considered to be one of the essential amino acids. What we mean is if it's an essential amino acid, we can't synthesize it. We have to eat it. But bacteria have this tripoperon. So all of the enzymes, A, B, C, D, and E, that are needed to synthesize tryptophan actually are part of this operon so that they can be synthesized together into a common messenger RNA that then can be translated into the 5 different proteins, the 5 different enzymes. So it's a very coordinated production of the enzymes when the bacteria senses, oh, I need to make some more tryptophan. So considering this operon format here, how many promoters are there for the tripoperon? Yeah, there is 1 promoter. How many transcription stop sites? 1. So there's 1 start site, so polymerase will bind to the promoter, transcribe all the way through. Here's the termination site. It will stop. So 1 start site, 1 stop site. How many mRNAs are made? There's 1. The same one can be made over and over again, but there's 1 species of mRNA that encodes for all 5 of these proteins. And so I just told you the answer. How many proteins are made? 5, exactly. So what happens here is that there are start sites for the translation machinery to hop on, synthesize protein E. There's a stop codon, it leaves. A new ribosome comes on here, synthesizes protein D, and so forth. That is what we mean by an operon. And that's in contrast to genes that are separately expressed, which is the way it is for many eukaryotes like our own genome. So it turns out that yeast, so single-cell budding yeast, they also are able to make tryptophan themselves. They encode all of the enzymes to make tryptophan, but they encode them on different chromosomes, different locations of chromosomes. They are not arranged in an operon. So in this case, still 5 enzymes. How many promoters do we need? Yeah, 5 different promoters. You need one here, one here, one here, one here, one here. How many stop sites? 5. They each have their own individual stop site. How many different mRNAs? 5. How many proteins? 5. So does everyone understand the difference between having separated genes and an operon? All right. So now let's move on to a new experiment where we're going to talk about how, suppose I wanted to know, is my yeast culture expressing these tryptophan messenger RNAs? Are they going to get ready to make tryptophan? Does anyone know the name of an experiment you can use to specifically detect a type of RNA? It is a northern blot, yes. So northern blot, that sounds similar to a southern blot, which we talked about a couple weeks ago. So in a southern blot, what we were doing is we were using a specific probe to detect a DNA sequence. For a northern blot, we are using a specific probe to detect an RNA sequence. The reason a southern blot is called a southern blot is because the scientist who figured out how to do that was named Ed Southern, so he named it after himself. There is no Professor or Dr. Northern. They just named a northern blot because in principle it is similar to a southern blot. There's also a thing called a western blot. What does that detect? Protein, exactly. So it's the same idea, just going with the direction. So, all right, so let's talk about how we would do a southern blot. So the first thing we would need to do is isolate our RNA. So if we're looking at our yeast cells, we want to see if a tryptophan messenger RNA enzyme is being made. We would collect all the RNA from our yeast cell. The next step is we denature that RNA and run it by electrophoresis through a denaturing gel. So when we were doing a southern blot, we needed to use restriction enzymes to cut our DNA before we ran it into a gel. Why aren't we using restriction enzymes here? Yes? So the RNA themselves are nice sized pieces. Most of them are going to run between 500 and 10,000 nucleotides long. So they already are cut into discrete pieces. The other reason is, do restriction enzymes cut RNA? No, restriction enzymes recognize specific DNA sequences, double-stranded DNA sequences, and cut DNA. So we don't need the restriction enzymes for our northern blot. It already will nicely separate in our gel. But then the next step is we just need to transfer that pattern of RNA. We need to transfer the RNA onto nitrocellulose, fix it so it's stably there. We then design a probe to be able to detect that specific RNA we're interested in. And what are the 2 things we need to think about when designing our probe? What does our probe need to be able to do? If we want to detect that tryptophan RNA, what should it do?
Should it be able to bind to the RNA, but also be able to detect it?
Exactly. Those are the 2 things. You design it to be able to base pair to the sequence that you're interested in, and you put something on it like fluorescence or radioactivity in order to be able to detect it at the end. Question?
Yeah. What denaturing are you doing to the RNA?
Yeah, so good question. So RNA, single-stranded, right? Do you think it ever folds back on itself and base pairs to other RNA species in there? So yeah, RNA, although it is transcribed as a single strand, it has a lot of double-stranded nature. And so you do need to denature it so that it will run as just a single strand in the gel. Mm-hmm.
Is that with heat or is that like using proteins?
We usually use heat and some chemicals to keep them denatured. All right, here's a question for you. So the probe, that sequence that we designed to be able to base pair to the RNA, does the probe need to be made of RNA, DNA, or either one? If we want to detect a band of RNA, would DNA base pair to it? Yes, absolutely, right? Would RNA base pair to it? Yep. So you could use either. This is true also for your southern blot. If you want to detect a sequence of DNA, a DNA probe would base pair to it, an RNA probe would base pair to it. It's your simple base pairing properties. Does that make sense? All right, so let's look at an example of an actual northern blot that a student did in my lab several years ago. So we use C. elegans worms in the lab to, in this case, study a gene involved with development. And so she wanted to know, when does this gene turn on in development of the animal, to get a better idea of when it might function. So she isolated total RNA from eggs, as well as after they've hatched into the larval stage, and then she denatured that RNA, ran it by electrophoresis through a gel. So it's going to be all the RNA that's present in the worm is going to be on the gel. It is going to get transferred onto nitrocellulose, fixed. She designed a specific probe to base pair to her RNA sequence of interest. She put radiolabel on it and then detected it. And here was the result. So what can you conclude from this result? You conclude anything from this? Is the RNA expressed in eggs and larvae and both and none? Where do you see the probe? Yeah, you see this nice big band here in the egg. You see nothing in the larval stage. So that means there was RNA expressed in the egg that base paired to the probe. However, there's another possible explanation for why nothing might be detected in the larvae. What if she forgot to load that lane, or the RNA got totally degraded? You would also see that result as well. So when we do these northern blots, it's always very important to include detection of a control RNA. And so a control RNA would simply be an RNA that's likely to be similarly expressed in all cell types and all developmental stages. Can anyone think of a good example of a control RNA? Some RNA you expect to be in every cell type. Like a housekeeping RNA. Maybe something that's involved in metabolism. Even all your cells have ribosomes, right? So they all need ribosomal RNA. So ribosomal RNA, tRNAs, these are abundant RNAs that are there in all cell types, and so you should easily detect them. And so that is exactly what she did. She also used a probe for a control RNA and sees that the RNA she ran in her egg lane, she detects a band, and in the larval lane, she also detects a band. So that confirms that her RNA was not degraded, it was run to the same level. So when she doesn't detect the test RNA, she can then conclude that this RNA is not being expressed in these larvae or is detected at much lower levels. All right, so how do we probe for a control RNA? Well, it's very straightforward. So in your initial northern blot, you have designed a probe that will base pair to your RNA that you're interested in, the test RNA. And as we saw, like all the RNA from the organism is there, but you're only going to detect what your probe base pairs to. Once you've done that, you have your result, then you just need to remove this probe. How could you easily remove a probe that is base paired to the RNA on your nitrocellulose? It's base paired using hydrogen bonds. How do we break hydrogen bonds really easily? Heat, right. You sort of throw almost boiling buffer onto your nitrocellulose. That will wash off all of your probe. And then what can you do? Then you design another probe against your control RNA. You rehybridize, and then that might be a different sized RNA, and so that will show up somewhere else on your gel, and then, or on your blot. And then when you see the detection there, you can confirm that RNA was intact, it wasn't degraded, and it was actually run in that sample. Question? Sometimes you can do that if you're very confident in the sizes of your RNAs, but if you're unsure if your test RNA might be differentially spliced or a different size, you wouldn't want to complicate your results with having too many different bands. But sometimes you can do them both at the same time. Other questions? Okay, so here's another example of a northern blot, this time looking at beta-globin mRNA, where they have probed for beta-globin mRNA in cells undergoing differentiation. So we have undifferentiated cells, cells that have been pushed to differentiate 48 hours and then 96 hours later. What can you conclude from this northern blot? Mm-hmm. So it looks like there is essentially no expression of beta-globin here in our undifferentiated cells. We're not detecting this RNA at all. After they've been differentiating for about 48 hours, we start to detect it. And then at 96 hours, we see this huge amount of detection. What that simply means is if it's a darker band, that means more probe is there. The only way you get more probe there is if you have more RNA there. So the expression of beta-globin mRNA increases as the cells differentiate. We're assuming that there's a control that they also probed for what we call loading control, and that was expressed the same in all of these samples. Okay, so we see that beta-globin is turning on. So what predictions can you make about the chromatin structure of the beta-globin gene at these 3 different time points? What do you think the chromatin structure is like in the undifferentiated cells at the beta-globin gene? Yeah, heterochromatic. It's very condensed. The gene is not being activated. It's not being turned on. And then what's happening through differentiation? Well, if you are turning on gene expression, that must mean polymerase is now able to access this gene and start transcribing it. So you're getting to a more open or euchromatic status. And what experiment would you do to test that idea? A DNase protection assay, exactly. Okay, so in this last section, let's talk a little bit about control of gene expression in bacteria. Again, we'll review what we mean by activated versus repressed. And it's just as important to activate genes as to also keep them repressed when you don't need them. There's a lot of energy that goes into expressing genes. The E. coli genome is about 4 million nucleotides long. It has the potential to express about 4,300 proteins. Is it constantly turning on all those genes, making all those proteins in its very short life cycle? No, absolutely not. And so it's important to control gene expression for the circumstances that it needs those genes to be expressed. We'll talk about what we mean by sigma factors. These are factors that help RNA polymerase turn on the right genes at the right time. And we'll discuss just one example of regulated gene expression in bacteria, the very famous lac operon. Okay, so when we say a gene is activated, what do we mean by that? What essentially are we saying? It's expressed, it's turned on. RNA is being synthesized. Transcription is turned on. And so then it's the opposite, repressed. We're not making RNA anymore. The gene is not being expressed. If you're a bacteria cell, what would be responsible for changes in gene expression? So bacteria just live to divide, right? But can they always just happily do that? No. What do they have to respond to? Yes. Yeah, that's a great example. Temperature, any changes in environment, how much nutrients they have available. In fact, the lac operon is an example of nutrient availability that we'll go through. Multicellular organisms like our cells, we also respond to environmental changes, but a lot of our transcriptional programs are actually regulated based on development as well. Okay, so let's talk about sigma factors. So what sigma factor is it is a protein. As you'll see in the next slide, there's a variety of different flavors of sigma factors, and they are required for the RNA polymerase to initiate transcription. The sigma factor itself binds to the RNA polymerase complex, and it guides it to specific sequences at the promoter region of the gene to be transcribed. And so sigma is going to bind to the promoter sequences, and it also assists polymerase in separating the strands so that the template can be revealed. Once polymerase then starts synthesizing RNA, sigma can leave, go join another RNA polymerase complex, and start the procedure over again. So there are a variety of different sigma factors that then bind to different sequences to regulate different genes. So depending on what condition is happening. So for example, sigma 70 helps recruit RNA polymerase to many housekeeping genes. So like ribosomal proteins, RNA polymerase itself, the gene that expresses that polymerase. And it really likes to bind to genes that have this promoter consensus sequence. So what do we mean by a promoter consensus sequence? It simply means this is the sequence most favorably bound by sigma 70 in this case. So in reference to +1, where transcription would start, upstream of that, you want to have this sequence at the -10 region, and then this sequence at the -35 region. If there is a 1 or 2 nucleotide change in a consensus sequence, can sigma still bind to it? Usually it can, but is it going to bind as well, as stably? No, it's going to be a weaker interaction, but you can have variability in these sequences, and you'll see an example of that when we get to the lac operon. If your bacteria suddenly have iron and they want to uptake that, then this sigma will then come on and it will turn on these iron uptake genes that then have a different sequence at their -35 and -10 region that will then be recognized by this sigma. So these different sigmas help the bacteria turn on different genes depending on what their environment is. All right, so let's finally now review this lac operon as an example of how nutrient availability can regulate gene expression in bacteria. So the lac operon encodes 3 proteins that are needed for lactose metabolism. So this is going to be the LAC-Z gene, and then there's 2 other genes, and then they're going to be controlled by this promoter and these regulatory sequences. But to really understand how elegant this lac operon is, we need to first think about, well, why would a bacteria cell even want to metabolize lactose? Why would it want to turn these genes on? What is lactose? Yes? It is a sugar, right. So lactose gets cleaved by that first enzyme in the lac operon, LAC-Z, into these 2 molecules. What are those 2 molecules, or at least what is one of them? Glucose, exactly. So it gets cleaved into galactose and glucose. Really like to use glucose for all kinds of things in the cell. All right, so the whole goal of turning on this operon is to be able to metabolize lactose into glucose. So with that in mind, let's think about what level of lac operon transcription the cells would have when, for example, there's no lactose available. Substrate's not available. Would you want to turn on these genes? No, there's no point in turning on enzymes to metabolize lactose if you don't have the substrate. So that will be the repressed state. All right, some lactose comes into the cell, but they still have some glucose. So what level of expression of the lac operon would you want in this case? You have the substrate, but you also have the product. Maybe a little bit of expression, right? Because maybe you're going to run out of glucose, and then you really want to take advantage of the lactose you have. And then finally, you have no glucose left in your cell. You suddenly get a whole lot of lactose. You want to really turn on high expression of this lac operon so you can metabolize it. So let's look at how the lac operon is regulated. All right. So there is a protein called the lac repressor protein. And what this lac repressor protein does is it binds to a sequence called the lac operator that's in the promoter region of the lac operon. When the lac repressor is bound to the operator, it blocks polymerase and sigma 70 from binding to the promoter. If polymerase can't bind the promoter, are you going to get any transcription? No. The only way you can ever get transcription is if polymerase can actually bind to the promoter and start synthesis. So if there is no lactose around, repressor is able to bind to that operator and block the polymerase from binding. All right, now we have our next situation. So now we still have glucose, but now lactose has come into the cell. And what does that do? It turns out that this sugar, this lactose molecule, binds to the lac repressor protein. And when it binds to the lac repressor protein, it changes its conformation so it can no longer bind to the operator. All right, repressor's off the operator. So now the promoter is free for polymerase and sigma 70 to sit down and start synthesis. But the synthesis is actually just a low level of transcription. So why is that? Well, just to review, we have lactose, the sugar binding to the repressor. Repressor can no longer bind to the operator, dissociates. We have polymerase, sigma 70, recognizing those -10 and -35 sequences, bringing polymerase to the promoter, but we get a low level of transcription. And the reason for that is because the lac operon promoter sequence actually has what we call a weak promoter. The sequences deviate from the consensus sequence that sigma 70 likes to bind to. So, because there are these differences, sigma 70 kind of binds, but it's weak. Only now and then will it bind well enough to get this low level of transcription. So we're going to need help in order to hold polymerase onto this promoter and get a high level of transcription. And that help will happen when we now have been depleted of glucose. So there is a signaling cascade that happens when cells run out of glucose that results in the accumulation of a molecule called cyclic AMP. So it's an adenosine nucleotide monophosphate, because there's one phosphate, and it forms this circularization. So that's where the cyclic, or the C, comes from. This is called CAMP. What CAMP does is it binds to a protein called CAP, catabolite activator protein. And when it binds to CAP, it recruits it to the CAP site at the lac operon near the promoter. And what that complex then does is it holds sigma 70 and polymerase on that promoter stably, so then you can get high levels of transcription. So to get our highest level of expression when we have lactose has come into the cell, whenever lactose comes into the cell, it's going to bind to the repressor protein. Repressor can no longer bind to the operator. That frees up the promoter so that polymerase and sigma 70 can bind. But the only way it can stably associate is when we have cyclic AMP that binds to the CAP protein, that then binds to the CAP site. So that's a DNA sequence that's next to the core promoter, and that's going to stimulate the RNA polymerase to allow for high levels of transcription. Any questions about that process? So think it through. Think about whether you want to express high levels, medium levels, or low levels of the lac operon. And finally, we're going to end with a clicker question to make sure you do understand this lac operon. And so here I've sort of drawn out the players. So these are the DNA sequences. What is this green box? What do we call the green box? It is the CAP site. It is the element or the sequence in the DNA that is bound by the CAP proteins. What is this orange box? It's the promoter that's going to have those sequences that sigma 70 needs to recruit RNA polymerase to bind to. They are not consensus, so it's pretty weak binding on its own. And what is this yellow box? It is the sequence that the repressor binds to. It's called the operator. So lac repressor will bind there, but only when it's not bound to lactose, right? So based on the above northern blot experiment analyzing LAC-Z mRNA expression from E. coli samples grown in different media conditions, which statement is supported by the data? Don't leave yet. You all want to beat the storm? Is it still raining out? Yeah, it's probably still going to be raining out. You can leave if you need to. I'm just kidding. But we will go over the answers for those of you who want to stay. All right, put in your final answers. All right, we have a slight majority liking B. Not a whole lot for E, but we have definitely answers or votes for all the rest of them. So let's go through this. Who wants to volunteer their answer? Yes, Lydia. All right, so we're going with B. The cap site is more likely to be bound by cap plus CAMP in sample 2 than in sample 1. So we only need to focus on sample 2 and sample 1. Why did you like that answer?
I like that answer because, well, the RNA is the control. So and then number 3 is obviously not very working, so I didn't. And then I look at the LAC-Z mRNA. The LAC-Z mRNA will only be expressed if there's lactose present. And then it's a really strong band, so that means it's transcribing really well. So then in order to have active transcription, we need the CAMP.
Yeah, so the first thing she did, which is important, she looked like, what does our control look like? So the ribosomal RNA should be equally expressed in all of our cell types if we did everything right. And that's looking good for samples 1 and 2. And then I just want to compare 1 and 2. So when CAMP is bound, we said that that recruits the polymerase, holds it there more steadily, so we get high levels of expression. We see much more probe bound in sample 2, which means there's much more of this LAC-Z mRNA being produced. So that would be our higher level of expression than what we're seeing in 1. So yes, B is the correct answer. But let's look at why the rest are not correct. A says condition 3 results in the least LAC-Z mRNA. Well, I see no LAC-Z mRNA. Why is that one not correct? What's my problem? The control. Oh, we can't really conclude anything here. Something happened to my sample. So A cannot be concluded because our control has a problem. C says the LAC operator site is likely bound by the repressor in condition 2. When repressor is bound, do we get any expression? No. If the repressor is bound, doesn't matter if you have CAP, doesn't matter if you have CAMP, you're not going to get polymerase bound, and you're not going to get any expression, so there's nothing for probe to bind to. That's the opposite of what we're seeing. In 2, we're seeing a lot of expression. D says the sigma 70 protein is more likely bound by lactose in samples 1 and 3, but not 2. Does sigma 70 get bound by lactose? No. What does lactose bind? The repressor, exactly. So that statement's just wrong. Sample 2 has higher glucose levels than sample 1. If we have high glucose levels, what do we have low levels of then? CAMP, right? And we need CAMP to bind CAP to really recruit polymerase to the promoter. And since we're seeing the highest level in 2 compared to 1, that can't be correct. It would be the opposite. It would be sample 1 would probably have higher glucose than sample 2. Does that make sense? All right, that's it for today. Good luck in the weather. Sorry for the technical problems.